My topic is biocommunication in the oceans, and I will be covering this topic into 4 modules. Module 1 consists of how a cell responds to different endogenous and exogenous factors, which were the first life forms to evolve on earth, and pathways of communication in prokaryotes and eukaryotes. The communication between the cells or organisms in most of the cases is facilitated by different chemical molecules called as semiochemicals. These semiochemicals, they try to change or modify the behavior of a cell in either way. Broadly classified into 2 types. One is pheromones, in which the chemicals operate intraspecifically, that is among the members of the same species, whereas in case of allelochemicals, the chemicals operate interspecifically, that is among the members of different species. Such a biocommunication leads to coordination in a behavior or a biological response. So, cells communicate with each other, as I said, by sending and receiving the signals, and these signals are either endogenous or exogenous signals. What are endogenous signals? These endogenous signals are the ones by which the cell performs its day-to-day activities like synthesis of enzymes or metabolism, etc. And this is mainly coordinated by the nucleus. Whereas, in case of exogenous signals, these are the environmental signals, and these environmental signals are very important for the cell to understand what is happening in the environment, and these endogenous signals, they trigger the biological response by interacting with the receptors that are present on the cell, or it has to enter the cell to and interact with the nucleus to trigger a response. This response may be the change in the shape of a cell, or it can be the secretion of some enzymes or the pheromones that are required for some reproduction purpose. So, this is a coordinated behavior, in fact, wherein the exogenous signals are pursued by the endogenous signals, and then there is a biological response is triggered. But it is very important to understand that these signals are very specific, and receptors to receive these signals are also very specific. So, you can see here that each receptor is specific for each signal, and there are different types of mechanism by which it can be interpreted by the cell. From an evolutionary perspective, it is possible to say that through convincing fossil records, the first type of cells to be evolved were morphologically similar to modern bacteria or prokaryotes. When the life on the earth evolved some 3.5 to 4 billion years ago, the first type of cells that evolved were the prokaryotic cells. So, these prokaryotic cells, which are the unicellular cells, then evolved into multicellular cells, that is eukaryotic cells. The primitive bacteria, or which we call as the old bacteria, they actually inhabited the most extreme environments, where the environments were acidic, less or low in oxygen, with no oxygen sometimes, and then they slowly evolved into eubacteria, or the most recently evolved bacteria. These eubacteria then evolved into eukaryotic organisms some 1.5 to 2 billion years ago. Now, you can see that this bacteria or prokaryotes are much different from the eukaryotic cells. Now, eukaryotic cells such as plant cells, and you can see here plant cell, and you can see here an animal cell, they are much different from the prokaryotic cell, that is bacteria. Now, in case of eukaryotic organisms, there is a true nucleus, and this true nucleus is separated from the cytoplasm by means of a nuclear membrane. Also, there are many membrane bound organelles which are present in eukaryotes, but the same is not present in the prokaryotes. Here there is no nuclear membrane. The size of these cells also are very small. These bacteria are around 0.2 to 2 micrometer in diameter, and in case of animal or plant cells or the eukaryotes, it varies from 10 to 100 micrometer in diameter in size. One important thing, or very important thing you can see here, is these 2 organelles which are here are very different from the rest of the organelles. First thing is that they are membrane bound structures, and it seems that they are the remnants of bacteria. Now, it seems that these primitive eukaryotes, they engulf these bacteria by the process of endosymbiosis. Now, if they are similar to bacteria, then there should be some similarities which shows that they are similar. Yes, there are some similarities. For example, they have their own DNA, they have their own ribosomes and they have the same bacterial configuration or function. Their size also you can see it is almost similar to that of a bacteria. They produce their own proteins like that of bacteria. Now, in this case you can see that the chloroplast, it has the chlorophyll, metabolism and enzyme similar to that of cyanobacteria, which was a primitive bacteria. And in case of mitochondria, you can see that the metabolism of this particular bacteria is similar to that of the respiratory bacteria. This shows that these particular remnants which we say of bacteria, that is mitochondria and chloroplast, is obtained by these eukaryotic cells by this process of endosymbiosis, and this is the first signature of biocommunication in the history of evolution. Now, the biocommunication then further evolved through a period of time through gene transfer. There are two types of gene transfer, one is vertical and horizontal. In case of vertical gene transfer, the reproduction is by cell division, and here it is mainly due to reproduction, that is the DNA is transferred from the parents to the offsprings. But in case of horizontal gene transfer, there are transformations, transductions and conjugations, and this is an important force modulating evolution in the prokaryotic as well as eukaryotic world. So, that means these cells are continuously exposed to different types of chemicals or the chemical signalings. In case of bacteria, the pathway of communication or the mode of communication basically is termed as quorum sensing. Now, quorum sensing is a mechanism by which bacteria talk to each other. They talk to each other by means of chemical signals, and they then coordinate and they function as a multicellular organism. This is also called as cell-cell communication or cell-cell signaling. Now, in case of gram-negative bacteria, they secrete these chemicals are called as autoinducers. In case of gram-negative bacteria, they are called as acyl homoserine lactones. So, what happens is these bacteria, they secrete these autoinducers or acyl homoserine lactones, and in the environment, and the other bacteria of the same kind identifies this signal and again secretes the same type of a signal. So, once this signal accumulates and reach a particular threshold, then it gives in the expression of some genes and some processes are gets expressed. That means they are achieved only at, as I said, it is achieved only at higher cell densities. In case of gram-positive bacteria, there are peptides which are also similar, which are also molecules like acyl homoserine lactones in case of gram-negative bacteria. Now, this quorum sensing is not only between the same species bacteria, it is also between different species of bacteria. And this type of quorum sensing is called as quorum sensing crosstalk. This is very important in the microbiology, or it has very major implications in the microbiology, because most of the bacteria in the environment or in nature, they do not exist as single species, but as multi-species films as that in the found in the biofilm. So, examples of some quorum sensing control processes include bioluminescence, biofilm formation, virulence gene expression, sporulation and competence. The pathways of communication in eukaryotes is much more complex when compared to that of bacteria, because they are multicellular organisms, and unlike bacteria where the cell interacts and the response is seen, in this case there are many cells and the cells sometimes need not interact with each other, but the cell has to secrete the substances in the extracellular environment and then it has to reach the target at distant places. There are 5 categories of chemical signaling found in multicellular organisms. First one is autocrine, in which a cell targets itself. That is a cell, this is basically seen in apoptosis or programmed cell death, where the cell produces a compounds and in turn that kills that same cell. The second type is direct, in which a cell targets a cell connected by gap junction. This is between the neighboring cells. The chemical molecules are produced by one cell and they pass on to the other by means of these gap junctions to create a response. In case of paracrine, a cell targets a nearby cell, that is, a cell secrete the molecules in the environment or in the space, and then they percolate or they reach the target cell. This is how the sensory cells function. And in case of endocrine cells, a cell targets a much distant cell through the bloodstream. For example, hormones which are released directly into the bloodstream and then they target the cell at distant places. Other than this, pheromones are also secreted or exuded chemical factors that trigger a social response in members of the same species. So, with this I come to the end of module 1.